All right. Good afternoon, everybody. Welcome to another installment of the Immuno webinars. We are thankful for Dr. Gilroy for accepting our invitation to talk on this specific date and for accommodating the students from the Lipid Mediators graduate course that are also joining us today. Thanks also to Dr. Daniel Gomes from the Federal University of Espírito Santo, who made the initial invitation to our presenter today. Derek Gilroy obtained his PhD from the William Harvey Research Institute, University of London, and did his postdoctoral training jointly at University of Houston and at Academia Sinica. He then returned to the William Harvey Research Institute. In 2003, Derek was appointed as lecturer and then senior researcher, funded by the Wellcome Trust at the University College London. In 2010, he was promoted to professor of immunology and is currently head of the Department of Aging, Rheumatology, and Regenerative Medicine. Professor Gilroy has won multiple prizes and honors, including the Bayer International Young Investigator Award, the Novartis Awards, and Peter Dressel Awards. And he's also a fellow of the British Pharmacological Society. He has developed several rodent and human models of experimental inflammation to investigate how the body regulates immune responses to infection, and when this goes wrong, how chronic inflammatory diseases may develop. He's the founder and the CEO of Penta Therapeutics, which is built on the premise that signals from chronically inflamed tissues suppress pro-resolution pathways and perpetuate chronic inflammation. I'd like now to thank Dr. Gilroy again for joining us today and cede the floor for his presentation. Dr. Gilroy, please.
Wonderful. Thank you very much, Daniel, Bruno, and everyone else for the opportunity to present our data here today. I've got many Brazilians who've come into my laboratory. Many have remained friends after they've left, which is always a good thing. And of course, Daniel is a long-term friend. I've a tremendous fondness for the Brazilian community, particularly the scientific community. So it's a real pleasure for me to share my data, our data, with you today. As the title said, it's going to be on epoxy oxylipins, human inflammation, and mononuclear phagocytes. Oops, there we go. Righty. So, summary of the talk. What are we going to talk about? A bit about inflammation and resolution of inflammation, of course. And within that context, mononuclear phagocytes, in particular monocytes, and their differentiating into macrophages, and the role that epoxy oxylipins play in this role, or in this setting, if you like. And then touch upon how this function of mononuclear phagocyte differentiation may contribute to impaired healthy aging and chronic inflammation. And to start with, when I'm always drawing up what I think inflammation is, it's inflammation on the y-axis and time on the x-axis. And by inflammation, of course, we always lump everything together. We throw heat, redness, swelling, pain, and edema all into the one container, just to emphasize the events that occur when we get an infection or an injury. But neutrophils are typically the first line of response to an infection or an injury, and followed by macrophages. Now, that can either be from tissue-resident macrophages or those that have infiltrated from peripheral blood. But by and large, their role is to eat up the apoptotic dead cells and help to hoover up and clean up the site of inflammation. And that happens within, as you know better than I, about 24 to 48 hours. And then when we're talking about acute inflammation, we oftentimes forget that it really is only the start of a more orchestrated, sophisticated, long-term process that develops long-term memory against that infection in the first place. And part of that process is how dendritic cells then speak to T cells that come into the tissue. And ultimately, it really is three things as I see it: pathogen eradication, the resolution of the acute inflammatory response, but leading ultimately to tissue memory. And part of that process really is not just the local tissue inflammation and events that are occurring there, but also much more quietly within the tissue and the draining lymph nodes, as I've depicted down here, as I say, that orchestration of the long-term adaptive immune response. And the other bit that we kind of forget a little bit about in inflammation is the role of the microvasculature that delivers immune cells to tissues, the fact that it swells, becomes red, becomes hyperdilated. And that really is the redness that we see at sites of inflammation. We know a lot about what causes that to become red and allow edema formation, but we don't know an awful lot about the resolution cues that switch off the redness and turn off that hyperreactivity in the vessels. And the other thing we don't really know an awful lot about either is how the nervous system within an inflamed tissue communicates with immune cells. That's something that we really need to get a greater understanding on. So I think as I see some of the key gaps in our understanding of resolution biology is the division of labor that functionally exists between myeloid cell populations, the crosstalk really between innate immunity and adaptive immunity, and also the crosstalk between immune cells and stromal cells within the inflamed resolving tissue and the microvascular endothelium and the pericyte within that. But also how pro-resolution signals dampen pain, plus other things that I'm sure you can add to the list. And also to layer on top of that, how things like what's now called the exposomes, external stimuli that come in and negatively affect our immune responses. And this age is an inherent thing, but pathogens, pollution, and genetic diversity and dysregulation can affect resolution of inflammation. All of them can contribute to chronicity of the response. But for today's talk, as I said at the start, we're going to talk about mononuclear phagocytes and the role that a family of lipid mediators called epoxy oxylipins play in this process. Now, a talk on lipids wouldn't be complete without a nice complex biochemical pathway on your screen, which is here. And just drawing your attention to the top polyunsaturated fatty acids that you know much better than I do: linoleic acid, arachidonic acid, DHA, and EPA. These are all called polyunsaturated fatty acids. Now, as you know, they can all be metabolized by the cyclooxygenase pathway to give rise to the prostanoids, the lipoxygenase pathways to give rise to the leukotrienes, or in this case, the point of my presentation today, they can be metabolized by the cytochrome P450 epoxygenases, mostly speaking, CYP2J2, 2C8, and 2C9, to give rise to what are collectively called epoxy oxylipins. So, for example, linoleic acid will be metabolized by cytochrome P450s to 9, 10, and 12, 13 EPO. Arachidonic acid, however, to 5, 6, 8, 9, 11, 12, 14, 15 EAT, and so forth. These lipid mediators are synonymous with driving pain, various aspects of inflammation, and vascular function. They're very short-lived, and they are broken down by a soluble epoxide hydrolase to inactive dihome or diheat metabolites. So, for instance, following this through, linoleic acid is metabolized by cytochrome P450s to 9, 10, or 12, 13 EPO. Very short-lived, act on the vasculature and other aspects of the tissue, and these collectively are broken down very, very quickly by soluble epoxide hydrolase to the dihomes, and the same for EATs to diheats. These are largely considered to be biologically inactive. Okay, and doing this, and what I'm drawing your attention to today with this very discrete red box around linoleic acid and arachidonic acid in particular, because that's where the story is going to be built upon and presented today. And also in that setting, very discreetly hitting the area, this big red arrow pointing specifically towards soluble epoxide hydrolases. And in truth, what we're doing is we're introducing an inhibitor for soluble epoxide hydrolase so we can imbalance one lipid mediator to the other, and with that information infer what these lipids are doing. Or presented another way, same story but in a slightly different presentation, what is really important to know Again, you know it better than I. Linoleic acid and arachidonic acid are present. Metabolites by cytochrome P450s, the EPOMs and the EATs are constitutively expressed in plasma, much higher levels than the prostanoids, and they're broken down very quickly by the soluble epoxide hydrolase to give the dihomes and diheats. And you can look at them as a sort of a balancing act. The EPOMs and EATs are slightly higher in concentration to the dihomes and diheats. And if we block soluble epoxide hydrolase, as per the previous slide, then you elevate the EPOMs and EATs because you back up the catabolism and you reduce the dihome and diheats. And therefore, in the mouse or in the human system, using soluble epoxide hydrolase inhibitors, if you have them, are a good way to begin to understand the relative role of these lipid mediators versus these. And that's what we did a couple of years ago with my good friend Dave Bishop-Bailey, in which we used a mouse model of Zymazan-induced peritonitis, and we gave mice a soluble epoxide hydrolase inhibitor, and we found that the pathway really did significantly affect, one, the trafficking of monocytes into tissue, and the manner by which they could eat up dead cells. Now, that was done in mice a couple of years ago, and since then, this drug, GSK 2256294, which is being proven to be safe in humans, could also be used not just in the mouse model, but also in our human models of inflammation, which I'll get onto in a minute. So whilst we had done this work and proven some of the role of these lipid mediators in mice, we now wanted to see what it does in our human skin challenge models of inflammation. Now, the problem, one of the reasons why we moved away from mice to human is that in humans, they've got 57 functional CYP genes, whereas in mice, they've got 102 functional CYP genes. So straight off the bat, you could say that humans are mice in terms of cytochrome P450s are really quite different. And the other thing is that the monocyte profile in humans can be really quite different under inflammatory conditions compared to mice. So if we take human blood monocytes, exclude out the T cells, focus on HLA-DR, and then gate on CD14 and CD16, you get this classic, what's called a waterfall profile, where we have classical monocytes here, which are CD14 positive, intermediate monocytes, which are double positive, and non-classical monocytes, which are CD16 positive. And generally speaking, in humans, in our resting blood as we sit here and speak, about 90% are classicals and about 5 and 5 are intermediates and non-classicals. When we give inflammation to humans, or if you get a flare of a chronic inflammatory disease like lupus or rheumatoid arthritis, you get what's called an expansion of these intermediate monocytes. So in reality, what happens is the classical monocytes differentiate the intermediate monocytes and they increase in number and in proportion. And we know this happens when we treat our volunteers with a skin challenge bacterial model. In mouse, we have a slightly different gating strategy to identify blood mouse monocytes. We gate on LY6C against CD11B, and you get what's called LY6C high, LY6C intermediate, and LY6C low, well known in the field. But in our mouse model, if we give the same amount of bacteria to mice as we give to humans, we really do not see an expansion of the intermediate monocytes. And when you consider, when you correct that for the amount of bacteria per kilogram, then in reality, we're adding in almost, this is 215,000, this is 500 million per kilogram. You really do not push the dial towards expanding numbers of intermediate monocytes. In the mouse model, you got to go 10 times higher, going up to 23,000 times the amount per kilogram in mouse than in human, in order to get a substantial increase in intermediate monocytes. So for that reason, we decided that we would not do any more studies in mice, but we would turn to our human skin challenge models to understand the role of intermediate monocytes or mononuclear phagocytes more broadly, and the cytochrome P450s in that system. So this is our human skin challenge model. We do a laser Doppler across the forearm to look at blood vessel dilatation, and that didn't work very well. And then we laser Doppler, so that gives us a baseline level. That's what it looks like on screen. And then we take blood first thing before we do anything else, get a baseline blood profile. And then we measure 7 centimeters from the crease of the arm. And then we put in our 15 million bacteria, which doesn't really hurt. After 4 hours, 6 hours, and 24 hours, you have a really nice redness. And then we extract out the cells using a negative pressure suction cup into a little blister. We pop the blister, extract out the cells and fluid, and measure whatever you want to measure on it. And this model is very quantitative, allows us to quantify the redness that occurs, vascular dilatation, using the laser Doppler, and by taking the fluid and cells, we can measure all the soluble mediators, lipids, and proteome. We can do biopsies, as you'll see later. And we do a lot of flow-assisted cell sorting using spectral flow cytometry. And so although it looks like a relatively small amount of fluid, it's actually very concentrated and there's sufficient quantities to do what we want on it. So using this model, this was our dosing strategy. Bit complicated, but bear with me. We took volunteers and we gave them the drug GSK 2256294 2 hours before we put in the bacteria. And we had baseline blood and we had blood 2 hours after the drug went in. That was before bacteria went in. So we got an understanding what was the impact of the drug on baseline epoxy oxylipins before inflammation. That is really important. And we do that because unlike the prostanoids, the epoxy oxylipins are relatively high tonically, constitutively in the blood. And so if we were going to do something to that pathway, we needed to know what the baseline effect was. So drug 2 hours later, took out blood again and looked at what the effect of the drug was on the relative ratio of lipid mediators. And then we came in intradermally with UV killed E. coli, and we looked locally at 24 hours, in some cases 48 hours, and matched blood samples. So we had events that occurred in the inflamed tissue, cells and fluid and redness and pain and heat, and we had matched blood samples. Okay. 2 people who carried out this were Olivia Bracken. She was the main postdoc working on it alongside Paranaz Jalali. Now, as I say, our model is very, very flexible. It allows us to measure lots of different things, both salient features of inflammation and events that are occurring in the tissue and in blood. And the first thing is that if we look at the development of pain in response to bacteria that goes into the skin, you can see, as you might imagine, this is the untreated volunteers. It increases and peaks at 24 hours. With the drug on board pre-dose, you can see that it caused an accelerated resolution of pain. And in the field of epoxy oxylipins, this is not an unexpected finding. One may have expected a more dampened, muted response at 4 hours. We got a more accelerated resolution of pain, if you like, with the drug on board. But looking at heat and edema, the amount of fluid that leaves the blood vessels and enters into the tissue, the drug really did not have a great deal of effect on those parameters of inflammation. Redness is another characteristic of how inflamed and vascular dilated the site is. This is what it looks like under Doppler, and pretty quickly you can see no massive difference, and quantitatively you can see no huge effect there. So in summary, so far, salient, superficial clinical features of inflammation, the big obvious effect was accelerated resolution of pain. Then the next question is, well, what happens? And our model is, you could almost say, a 2-compartment model. Blood is one compartment because the injection of the bacteria into the skin triggers a mild, but nonetheless distinct systemic response. and of course, events that are occurring within the injected tissue, as you might expect. So, what was the events that occurred in blood over time? Now, this is really important. I emphasized how we gave the drug, 2 hours later, took blood, and after that, we then injected the bacteria, again emphasizing that we were very keen to see what impact the drug has on lipid mediators before any infection goes in. Out of all of the lipid mediators that it could have elevated, we did a very broad spectrum lipidomic analysis with Darrell Zeldin and Matthew Eden at the National Institute of Health in the United States. And very distinctly, two things happened. 12-13 EPOM from EPA went up, eicosapentaenoic acid, and 14-15 EAT from arachidonic acid went up, as we might expect. And although not included here, the corresponding 12-13 dihome dropped down, and the corresponding di-EAT also declined. So we got a very clean inhibition of soluble epoxide hydrolase, elevation of these key EAT lipid mediators. And then throughout the inflammatory response with the bacteria on board, we can see that these lipid mediators are elevated in blood, almost as you'd expect. So that was the lipids that went up in blood. So what? What happened from an immunological viewpoint? Now, we had no real preconceived hypotheses. Maybe we were looking at events within the myeloid compartment, and we have some interesting data also on neutrophils here, but we took a very agnostic approach. I like to call it beat the bejesus out of the tree effect and see what falls out. In other words, design your panel broad enough such that when you look in spectral flow cytometry, you can find something interesting. So you're agnostic about what you're looking for. It's a very sophisticated approach, of course. In any event, just to remind you about blood monocytes, because this was the blood compartment that I'm presenting on today and in which we had seen a significant effect on. So as I said earlier on, human blood, you got 3 classical monocytes, intermediates, and non-classical, distinguished by the relative expression of CD14 and CD16. These have specific roles. Generally speaking, efferocytosis, intermediate monocytes, antigen presentation, and cytokines, and as you see later, potentially tissue damaging properties, which we're almost wrapping up a paper on that. And then the non-classicals involved in viral particles reactions. And again, you can distinguish them based on CD14 and CD16 expression. About 90% at any given time in our blood is classicals 5 and 5. Now, where these cells arrive from is quite important in the context of the data presented today. This is my good friend Simon Yona. He did this work in my laboratory a few years ago. He's now set up his own lab in Israel, and this is a beautiful, elegant experiment. And what Simon had done, he hypothesized that classical monocytes differentiate to intermediate monocytes, which in turn differentiate into non-classical monocytes in blood. So he took out this lot of cells here, classical, intermediate, and non-classical monocytes out of a volunteer. He put them into the machine to confirm that this is the pre-sort. He then sorted the classical monocytes, the CD14 positive, CD16 negative, put them back into the machine to confirm that this is what he was injecting into Mr. G humanized mouse. And what he had found was that after 10 minutes, again, CD14 and CD16 for orientation, these cells that he had just post-sorted to confirm what he was injecting, so he injected these cells into the mice. After 10 minutes, this is what they look like. The next day, this is what they look like. And then 2 or 3 days later, this is what they look like. In other words, they went through a cascade of differentiating from classical intermediates to non-classicals. And in addition to that beautiful, elegant experiment, he also did deuterium label of monocytes in healthy volunteers and mathematically probed the time frames and the relative quantities that classical monocytes differentiate into intermediate and non-classical. So that was a simple, differentiated linear molecule for the fate of monocytes in humans, naturally throughout the course of a day. So when we looked into the mononuclear phagocyte population, just to take you through this slowly, this is the untreated volunteers, drug on board, but starting from when the bacteria goes in to the arm, and this is in peripheral blood. And what we always see is a slight increase in the numbers, not proportions, and I'm always very careful that we always report on numbers, because this is a really key thing. There's always a slight increase in numbers of classical monocytes that sort of come back down after 24 and 48 hours. And we always see this very clear reduction, disappearance, you might almost say, of intermediate monocytes, but they bounce back after 24 hours. And this is kind of followed temporarily in a time-dependent manner, I mean, by the non-classical monocytes. And it kind of illustrates how one does differentiate into the other. Now, what happened to classical, intermediates, and non-classicals with the drug on board? Well, for classical monocytes, not an awful lot, pretty much the same as here. But what we really noticed that was distinctive was that the intermediate monocytes that would normally recover from 4 to 24 hours did not. And this is confirmed mathematically when we did statistics. And you could almost say the same for the intermediate monocytes. So you could say that at this point, there was with GSK 2256294 on board, it looked like there was some sort of a blockage in the differentiation of classical to intermediate monocytes. So we took some classical monocytes out. Remember, there was 2 lipid mediators that were elevated. There was EPOMs and EATs. And we took human monocytes, classical monocytes, put them in vitro, and we gave them 1 micromole of 1213 EPOM. And we found that after 24 hours that there was an accumulation of what looked like classical monocytes in vitro, a reduction in intermediates, and a slight trend towards a reduction in non-classical. So in vitro, as these cells naturally differentiate, this was blocked by 1213 EPOM, but the other lipid mediator that was elevated with GSK 2256294 on board did not affect that process. And so we could begin to get pretty sure that it was an effect of the 1213 EPOM on the differentiation and not the 1415 EAT. The other thing we looked at that could explain this phenomenon is that these classical monocytes differentiate in intermediates become sticky and stick to microvessels via the upregulation of cell adhesion molecules. We did extensive analysis of the peripheral blood mononuclear phagocytes following drug treatment, and we found no increase in the surface expression of cell adhesion molecules that might explain that they adhere to microvessel lining and sort of hide away from us. We do have results in neutrophils, which I'm not going to go into today. Stick with mononuclear phagocytes. And so the question then is, well, how potentially could 1213 EPOM be impairing the differentiation of classical to intermediate monocytes? And again, we did a lot of extensive look at pathways that are elevated in classical monocytes as they differentiate P38, PPAR gamma, SP1, and so forth. And the one thing that was really quite consistent was that as classical monocytes differentiate to intermediates to non-classicals, there's a decrease in P38 MAP kinase. So we did wonder if that played a role in the differentiation process. And we noticed that when we incubated classical monocytes with 1213 EPOM, that you got this significant inhibition in the phosphorylation or activation, if you wish, of P38 MAP kinase. And if you give losmapimod, which is a P38 MAP kinase inhibitor in vitro, to the monocyte differentiation experiment that I described a couple of slides ago, losmapimod does precisely what the 1213 EPOM does. Increases classical monocytes, decreases the differentiation in vitro to intermediate monocytes, and there's a sort of a trend towards a knock-on effect to the non-classical monocytes. And with Arnie Akbar and Emmett Chambers, we did a study some time ago in which we gave losmapimod to volunteers. This is the P38 MAP kinase inhibitor used in the previous in vitro experiment. And we asked, if we looked at the blood, what impact it would have. We would follow, therefore, that losmapimod would do the same as GSK 2256294. And indeed it did. So the summary of the data so far is 1, within the blood vessels and circulatory system, 1213 EPOM and 1415 EAT are present at about 1 micromole concentration. And via P38 MAP kinase that's upregulating these cells as to differentiate. 1213 EPOM blocks P38 MAP kinase and blocks the classic to intermediate differentiation pathway. So that's in blood. We get a reduction in these cells in blood. Well, what happens, therefore, at the site of inflammation? What information can we gather from there? Again, just to remind you, inflamed skin, matched blood, sites of inflammation, 4 hours and 24 hours. We did histology and don't bother looking too much at these details, only to tell you that we took biopsies at baseline for 8 and 24 hours, and we mapped the increase in expression of the cytokine P450s within the immune cells, increased elevation of soluble epoxide hydrolase following E. coli injection into the skin, again, primarily in immune cells. And the lipidomics on the blister fluid, we bundled for the purposes of today's talk most of the prostanoids, thromboxane, PGF2 and prostacyclin and PGE2, elevated around 4 hours, as is typically the case with prostanoids following injection of a bacterial infection into tissues. But the cytokine P450 epoxy oxylipins began to peak around 8, 14 hours and come back down around 24, 48 hours. And also, not just at the site of inflammation, in peripheral blood, but soluble epoxide hydrolase also elevated 1213 EPOM in the blister fluid. So at the circulatory system, but also at the site of inflammation, we can see the 1213 EPOM was increased by the inhibition of soluble epoxide hydrolase. But in comparison, controls 1415 EAT was not. So it's very consistent with the EPA-derived 1213 EPOMs. And it followed, therefore, that the amount of classical monocytes per blister versus the intermediate monocytes per blister, this population here, drug-treated versus non-drug treated, were significantly inhibited. This barely reached significance, which didn't correlate with peripheral blood. We got a massive inhibition in the expansion of the intermediate monocytes in blood. We got a modest, although significant, inhibition here at the site of inflammation. And I think one explanation one needs to remember is that in circulating blood, it is a massive pool for these cell types, whereas at the site of inflammation is comparatively small. So the real rate limiting factor is not necessarily a limiting number of cells in peripheral blood, but actually the relative size of the blood compartment to the inflamed tissue site, which is about the size of a dollar coin or something like that back in the day, quarter. So what we've learned so far is that in peripheral blood, again, remind you, 1213 EPOM and EATs constitutively expressed, playing roles in maintaining vascular health and well-being. Cytochrome P450 derived 1213 EPOM controls the natural classical to intermediate cell differentiation pathway. And that at the site of inflammation, not just do you get reduced intermediate monocytes when we block GSK 22569 soluble epoxide hydrolase in peripheral blood, but we also get a reduction at the site of inflammation of intermediate monocytes at 24 hours. The question is, well, what impact does that have on the tissue at 24 hours? Well, the other cell types that's present when intermediate monocytes are coming into the tissue are CD4 and CD8 cells, quite naturally, as acute inflammation progresses to adaptive immunity within the tissue. And what we found was that that the inhibition of soluble epoxide hydrolase reduced intermediate monocytes at the site of inflammation. It also reduced the numbers of CD4 T cells, Tregs to some extent, a little bit, not much of a significant effect on CD8 cells, and we got an increase in the amount of dead T cells that we could detect at the site of inflammation, and also markers of secreted cell death. So we were getting the impression that perhaps intermediate monocytes either act as a chemoattractant for T cells, or once T cells get into tissues, that intermediate monocytes keeps T cells alive. And given this data about the dead T cells and the secretion of danger signals, it's likely that intermediate monocytes support T cell survival and activation. And that's precisely what we found. That if we take out intermediate monocytes, incubate them with peripheral blood CD4 T cells, that you get an increase in cell activation markers CD25, 39, 69, and you get increased proliferation as well of T cells. So the model that we're looking at is that once intermediate monocytes leave tissues and leave blood and get into tissues, one of their role is transiently keeping CD4 T cells happy and in a state of activation to, if you wish, prime tissues for local tissue adaptive immune responses. So what happens when resolution goes wrong? Everything we described so far, resolution of inflammation, is behaving itself correctly. And there are many examples now in the literature of where intermediate monocytes in the blood of juvenile idiopathic arthritis, lupus, metabolic disease, cardiovascular disease are elevated significantly over long periods of time. And given their inflammatory pathogenic role, then it is not unreasonable to think that these cells could be quite dangerous during chronic inflammation. And Emma Chambers from Arnie Akbar's group some years ago, well, more recently actually, but she was in this group a few years ago, she has shown how intermediate monocytes are constitutively elevated quantitatively in the blood of old people as we get older. About 4%, as I've said before, in young people, doubles to about 9% in old people. And they are very inflammatory, making things like IL-6, IL-8, IL-10, and to a lesser extent, TNF-alpha. And some work that we've been doing in my lab with my good colleague, Dr. Mohammed Shipa. Again, if we look at the blood distribution of monocytes, intermediates, and non-classics in healthy people, 93.6. But in lupus, they increase from 3% up to 7%, and in active lupus goes up to about 12%. So a significant increase over quite a long period of time during disease flare. You get up to about 12-15% population of intermediate monocytes. And unlike any other cell in any other state, they are highly inflammatory, making lots of IL-6 and TNF-alpha, whereas the classicals and intermediates do not. And if we take out these intermediate monocytes, and we did some old ink proteomics on them, we can find that not just are they tissue pro-inflammatory, but they are releasing tissue damaging properties, granzymes, TIMPs, S100 proteins, and granzyme A and B, and granzyme H. So we're now excited in the idea that during chronic inflammation, you get an expansion of what we call intermediate monocytes that last for the duration of an inflammatory flare, which could be a couple of days up to a couple of weeks. These intermediate monocytes are enriched in not just pro-inflammatory, but also tissue damaging properties. We've done some experiments which were published in the paper showing that with T cells, intermediate monocytes are tissue damaging. And we really want to sort of uncouple the effects that are occurring in an inflamed joint during a flare, where you have inflammation, the heat, redness, swelling, and pain, which is of course salient and palpable when you're a patient with these sort of conditions. But what's going on inside there is this insidious, continuous erosion of tissue and breakdown of bone brought about by intermediate monocytes. And so we are now very keen on, we have a patent on the GSK 2256294 as a drug that will block the differentiation of intermediates to non-classical monocytes during inflammatory flare. And we're trying to set up a trial to ask whether that drug will dampen down the signs of tissue damage and tissue erosion during repeated flares in these disease conditions. So going to round up, just saying that a declaration of interest, CEO and founder of Penta Therapeutics, and an acknowledgement again, thank you to my good friends and chums over in Brazil for inviting me here today to talk about our research. A big acknowledgement to Olivia Bracken and Parinaz Jalali, Dave Bishop-Bailey, who got us into epoxy oxylipins in mice and then in humans in the first place, and Daryl Zeldin and Matt Eden for the lipidomics done on the samples. And a big thank you to you all. Thank you very much.
All right. Thank you very much. As always, we are going to open the floor for questions from the students first. So If there are any questions from students here, online or in São Paulo, please raise the hands physically or virtually. I don't know if there are any questions in São Paulo. Can see. But actually, I'm going to start this ball rolling with some questions of mine. And if people want to ask any other questions, please just call me up. So I was wondering, when we talk about polyunsaturated fatty acids, we always think about diets, right? And because we do need to get these fatty acids from diet. So I was wondering if you know anything about the diets of these volunteers and even patients that you looked at. And also, if the diet actually affects the expression of the cytochrome P450 enzymes, because they are also known to respond, right, to the substrates.
Correct. So we looked at the polyunsaturated fatty acid levels before and after in all of our volunteers, and even compared them to people who weren't in the study. And all the levels were pretty much equivalent across the board. Almost regardless of their diet. We took a quick look at their diet. We asked them to fill out diet cards, and there wasn't a huge difference. Some people were on more of a Mediterranean diet than others, but in reality, the the corresponding blood levels of polyunsaturated fatty acids were quite high, and were pretty much independent of diet, and they were there in such excess amounts it probably wouldn't have affected levels anyway. So that was the first question. And then the second was was it the expression of cytochrome P450s? Was that the question?
Yes.
So again, baseline levels in our skin, at least, were all pretty much the same, and they all elevated in response to the bacteria.
All right. Thank you. We have a question from Miriam. Miriam, please.
Hi, Derek. Thank you very much for your talk. I have a question on some of the side effects you mentioned, the pain specifically. Do you think that could be a matter of a different type of mononuclear cell there, like the difference between the classical and the intermediate, and something that they are producing and stimulating the pain receptors? Or do you think it's something direct and that could be used in other sources of pain, since the edema seems similar?
Yeah. Don't know, Miriam. We're not pain laboratories, just measured it because we could. And it's such a shame for us. We really do need to stain for nerve fibers to see where the immune cells are. I would not be surprised, to answer your question, really, if intermediate monocytes were found in close proximity to nerve endings in our model.
Mm-hmm.
Not surprised at all. Or perhaps release insoluble mediators. I'll tell you something else of interest. We did these models on patients with rheumatoid arthritis and osteoarthritis. We haven't published yet. We're waiting on some additional data. But would you believe that the site of bacterial injection in rheumatoid and osteo, they could not detect pain at all? Now, you'd expect it to be the other way around. Now, you might say, well, these people are so used to pain, they're kind of, sensitized to it. But what we also did was we had this gadget. It's kind of like you could say like a mobile phone, and had a prod on it. And you can push it into another part of your body to a certain pressure, whatever that pressure might be. And you then register pain. So you can apply the same level of local pressure to our rheumatoid and osteo as to our healthy controls. And at other tissues, they could experience the same level of pressure-induced pain. So it didn't look to us like it's got to do with the fact that they're just more used to pain. There's something quirky going on at the site of inflammation. And there's definitely a shift in these people in the amount of intermediate monocytes that happen at the site of inflammation. So that you might be right, there might be a link.
That's very interesting. But in case of the rheumatoid arthritis, the local joint pain did not change, right? It was only at the bacterial site of infection.
Correct. Yeah. So the rheumatoid patients, to be clear, their disease was pretty, you could almost say burnt out. They were off steroid, they're off treatments, they were sort of stable for quite a period of time. That was the condition for our study to be done. Whether it meant that one explanation is because they're in a permanent state of resolution, that there's sort of anti-inflammatory signals are activated while they didn't express pain. I don't really know. So they did not have joint pain at the time or within a year of being on the study. That was the agreement we had with ethics in order to go ahead with the study.
Okay, thank you very much.
You're welcome.
So keeping on this theme now of potential side effects that the hydrolase inhibitors may or may not have. Is there any concern about long-term treatment with these inhibitors and the potential to change these differentiation of monocytes and impair antiviral response, or even with the skin response to eventually modify vaccinations and other type of adaptive responses?
Yeah, so long term, you can be on these for a couple of weeks, and there's been some side effects with a slight increase in heart rate. So we're pretty tolerant to them. We would aim for people to be on them for no more than 1-2 weeks maximum, just to block the expansion of these pathogenic intermediate monocytes during inflammatory flare. Now, what we'd be aiming to do, if you wish to look at it, is restore homeostasis. In other words, reel back in the expansion of these intermediate monocytes, not knock them out, but bring them back down to a homeostatic level. We would not be aiming to impair baseline immunological processes, just dampen those that have gone out of control during the flare. So we would hope, therefore, not to compromise host defense.
Right, perfect. We do have a question from São Paulo. I don't know who will be asking. Hi, Diego.
No, it's me, Gustavo. Hi, Derek. Gustavo here. Thank you very much.
Sorry, it was such a small picture. I could not see you.
Right, now I'm here.
Hey, Gustavo, nice to see you.
Nice to see you too, Derek. Great talk. I wonder if you had the chance to investigate whether trained immunity would interfere with the metabolism of epoxy oxylipins and then the differentiation of monocytes, so on and so forth.
No, we haven't done that, Gustavo. We haven't done it. It's on our list to do, using BCG vaccine. It's like everything in this game. We have a long list of things we'd love to do, but time and money and resources are always the rate limiting factor. But I think it's a beautiful question, and we will love to do it.
Thank you very much. Great work.
Bruno, can I follow?
Oh, sure, please. Go ahead.
Question? So I don't know if maybe I missed it, but since these mediators are so high in blood and changes in the monocyte populations happen so quickly, did you happen to investigate anything, any sepsis model, or are you planning to do that?
No, we haven't. And again, like the trained immunity, it is on our list. They have been done in the past, the cecal ligation puncture model, and it was found to be protective, not particularly good at clearing the infection, but pretty good at stabilizing the cardiovascular and inflammatory response. The effects weren't overwhelming. I don't believe they impaired extended the life of the rats. I think the studies were done in significantly. So the effects in sepsis weren't overwhelming.
Thank you.
All right. We do have a question on the chat from Elvira. If I can ask this question, is about the mechanism of action of the soluble epoxy hydrolase inhibitors inhibiting P38 or on P38?
The mechanism, a good question. It's so what happens is these effects, when we put them onto cells, are very, very quick. I see there's another question about the neutrophils. Was that also by Elvira?
Yes.
I'll answer both at once. So we don't know is the answer. I think what happens is it binds to protein complexes in the cell very, very quickly. I think the site of action, these lipid mediators don't really have any recognizable classic receptors. And when we put them onto cells, the effects are very, very quick. So I think the lipids are acting on blocking the assembly and phosphorylation of complexes within the cell. And when we look at neutrophils, Elvira, we get a tremendously obvious effect on the aging process of neutrophils. And that was so profound, it was even more effective than the impact on the intermediate monocytes. We could, you know, if you want to jump on a call sometime next week, we can share our data. The effects of the pathways on the aging processes of neutrophils and the impact it has was surprising. And we're now trying to finish off the experiments, proving which lipid mediator is altering the aging of blood neutrophils and therefore their functionality. So yes, we had a big effect on neutrophils. Mechanism, again, very, very quick. I can only assume that the shift in lipids towards the EPOMs and EPOMs is acting on some sort of intracellular protein complex, either disassembling it or very rapidly helping to assemble it. But I suspect disassembling and blocking phosphorylation.
Right. Changing gears a little bit, since we have a big audience of people here studying lipid mediators now. Are these P450 mediators, are they dependent on phospholipase A2 activity and which phospholipase A2 activity? Because does not seem to have such a big market release like what's the very beginning that you showed for the PGE2, right? So they're much mild kinetics. So is there cytosolic phospholipase A2 alpha involved, or maybe IPA2? Which one is?
Yeah, that's a good question. In our model years ago, we found that earlier on in the response, it's cytosolic. And then later on, it was the inducible IPLA2 that released the second wave of prostanoids. In our system, we haven't had it. We can certainly, linoleic acid is definitely, you know, independent of that pathway. There's excess amounts of it around without cell activation. Of course, arachidonic acid is SN2 located and requires phospholipases, undoubtedly. So in part for the arachidonic acid, it's phospholipase driven. Probably linoleic acid, it's actually soluble present in the blister fluid.
Right. Perfect. Don't know if you have any more questions from São Paulo, online.
No more questions here, Bruno.
Okay, so I guess we are also done here. So we once again would like to thank Dr. Gilroy. Really nice presentation. It was really very exciting data, and we are looking forward for the follow-ups on all these data and to actually see these compounds to reach patients that sorely need of new medicines. So thank you very much.
My pleasure. To all in Brazil, have a great day.
You too. Bye-bye.